Hello, everybody. You guys are in for a treat today. We are going to take a look at Chapter 7, which is all about the functional organization of the nervous system. And it might just be me, but I think it has a lot of interesting points. So without further ado, let's get started with our lecture. We have mentioned the nervous system several times before. We mentioned it when we were discussing homeostasis in Chapter 1. We talked about it when we discussed communication patterns that the body can utilize. We also even discussed it in our last section when we were doing the sensory system, and we discussed the fact that the sensory systems will go ahead and respond to the stimuli, and once they have a change in their memory potential, they will go ahead and signal that excitation over to the afferent neurons, which will be able to communicate it with the integrating center of the nervous system. So we've kind of laid the base work already. We hopefully have an okay idea as to what the nervous system contributes. So what we'll do in this chapter is we'll take a look at the specific organs that belong in the nervous system. We're going to take a look at their anatomy and how they function. And of course, we'll do a comparison across different animal models as we peruse through. Now, before we get into the nitty-gritty detail, let's get some nice, a nice little overview by taking a look at the illustration that's on our slide. And what it's showing you is that when you look at the nervous system, you're basically taking a look at the combination of a sensory, an integrating, and an output type of pathway that have to communicate with each other. And along the way, there'll be different neurons that we'll be utilizing. And on our little graph, what we can see happening is that there are three main neurons. You have the afferent neurons, which will be responsible for being able to receive the stimulus from the sensory receptors and passing it along to the integrating center, where they will meet the interneuron. And the interneuron is almost like a little synaptic bridge, a little connecting point. It's going to allow the afferent neuron to bridge into the integrating center. And once the integrating center has decided what the method of action should be, it will go ahead and communicate it through the efferent neuron. And the efferent neuron, as you can see from our graph, will be connected to our effector organs. So that is then when you're going to get your final product. So maybe the muscle will contract or the glands will secrete. You'll start producing a certain hormone. You'll get the effect that the body was looking for. Within the nervous system itself, and when you do your comparison of afferent, interneurons, and efferent neurons, one thing I definitely want to point out is that the most common one that we have is definitely going to be the interneuron. That is the most common neuron that we have in our body. And as you can see from the graph, since it's correlated with the integrating center, that means that the majority of them will be located within the central nervous system. The central nervous system is basically the brain and the spinal cord. And then what we see happening is anything outside of that, so when we take a look, for instance, at our nerves, that would then be part of the peripheral nervous system. There will be lots of examples of how we'll look at the fact that the nervous system can be very complex and very coordinated, and we'll take a look at how some animals have a more advanced level of central nervous system than others. But there's also some examples where you have animal groups that really don't have a nervous system. And one of those examples are with the cnidarian group. This is when you take a look at animals like jellyfish or sea anemones. What you see happening over here is instead of using a central nervous system, they will actually use a nerve net. And the nerve net, if you can go ahead and take a look at your picture, will be more of an interplay of the different nerves. They will not necessarily have to be divided between afferent, inter, and efferent. In fact, what we see happening is a lot of these neurons will be what we call bipolar, meaning that they're able to receive both a stimulus and they're able to dedicate the effector that needs to be stipulated. Now, please don't underestimate the function of the nerve net, because it turns out that they're still able to do quite an amount of complex behavior when we take a look at our scenarios. In fact, I've embedded a little video for you that you can take a look at your leisure, where you can go ahead and you can see how your sea anemone will go ahead and have very complex swimming pattern and is even able to somersault. And I have to apologize because of my lisp. Anytime I say the anemone, the word just doesn't sound right. So my apologies. But hey, What can I do? Okay, it's my lisp. We also see that there's some terminology that will come into play when we take a look at mammals and other organisms that do have a complex nervous system. So I just want to make sure that we kind of recognize them and know what we're talking about. Some of these are very straightforward. So for instance, bilateral symmetry, that usually means that you have a specific right and left side. In our case, for the nervous system, they tend to be mirror images of each other, but this will come into play when we start discussing the fact that it's quite common for a signal to come into the right side and then cross over to the left side of the brain to be analyzed before we can decide what the effector needs to be. The nervous system also loves to do cephalization, which basically means that it will have a collection of sense organs that will be concentrated toward the anterior end. So this would kind of be the face and the mouth region. So many of us are thinking, isn't that where my brain's located? You would be completely correct in that. So there will be some type of integrating / control center. Ganglia is when we take a look at a group of neuron cell bodies. Nuclei is when we take a look at a group of neuron cell bodies, the same, but it's within the brain. So it's all about the location. And a brain, by definition, is going to be an integrating center where you now have the clusters of both the nuclei and input from the ganglia up to a certain point. And then the two last terms are tracts and nerves. Both of them will indicate the axon, and the axon obviously is the tail end part of the neuron. If the axon is staying within the central nervous system, then we call that a tract. If we see that it's radiating out of the central nervous system, then we're going to take a look at nerves. We also see that when we take a look at the structure of a nerve, we're just looking at groups of many different axons that are bundled together. And these will be often separated by different layers of connective tissue. So go ahead and take a look at your illustration, and let's start all the way on top. We're going to see our little axon right over here. So this is our tail end of our neuron, the one that will go ahead and send off the signal that the soma has decided on. And what you might notice on your axon is that you have sections that are myelinated. Myelinated means that it has a lipid wrapping around the axon. And it turns out that the myelination will actually speed up the nerve signal, so it's very beneficial. Not all axons are myelinated, but either way, you could see it's been illustrated in this example. Now back to our connective tissue. Around every single axon, you have an endoneurium connective tissue that will wrap itself around the individual axons. And now what we see happening is that your body will take the axons and it will bundle it together into a fascicle. And the fascicle will then get its own connective tissue sheet, and this will be the perineurium. Okay, so we have the endo for the individual, the perineurium for the fascicle, which is your bundle of nerves, and then last but not least, if we put several bundles together, that is then what we call a nerve. And the nerve will have an outer connective tissue layering called the epineurium. And notice how in the epineurium you will not only have different fascicles, different bundles, but you will also have the blood vessels that will be embedded within it. Now, when you look at a nerve, oftentimes it's classified as a mixed nerve, which means that within the different fascicles, you'll have a combination of both afferent and efferent neurons. So neurons that can reply to the sensory systems and neurons that can reply to the motor responses, so the effects that need to be accumulated. And in a little bit, I'm going to show you a picture of the nervous system across different animal groups. And we're going to take a look at how most of them will have that cephalization, that collection point where the sense organs are more towards the anterior end of the body. Obviously, the cnidaria, and in our case, also the echinoderms, the starfish, they will be a different bit of an exception because instead of cephalization, they'll be using their nerve nets and their bundles. One thing that I do want to point out is that what we'll talk about as we kind of progress through is that obviously there'll be a wide variety of complexity within nervous systems. And usually what we see happening is that the more complex the nervous system is in an organism, the more neurons it creates. However, it turns out that just because you have more neurons doesn't mean that you're better at integrating information or exhibiting more complex behavior. It doesn't matter about the number of neurons. What's really going to make the difference is the amount of synaptic connections you can make, because it turns out that all of your tasks, all of your memories, all of your skills are actually stored within these synaptic connections. And a synaptic connection is basically the point where the neurons are able to communicate with each other. And just to kind of give you an idea, it's estimated that the human brain has over 100 billion neurons that collectively can make over trillions of different synaptic connections. So it is a wide amount of information that can be stored, that can be learned, that can be expressed. So please keep an eye on that. It doesn't necessarily mean that if you have more neurons, it equals a higher level of intelligence or skill. It's all about being able to make more synaptic connections. And here is our illustration of the nervous system across different animal groups. And as you can see, all of them except for the highlighted A and G, the cnidarians and the echinoderm, they will have a brain, whereas A and G will go ahead and have their nerve net that they're utilizing. Keep in mind that with the nerve net, they're still utilizing the neurons. They simply don't have an integrating center towards the anterior end as what we see with all the others. So for instance, when you look at your alligator, you're going to see the mention of the brain by the skull. Even when you look at your crab, you can see it right over here. Even when we come over to our worms, we see that they'll have a collection of nerves that will be dedicated towards the brain segment. All right, here is a look at the vertebrate central nervous system. And two things I want to point out. First of all, we have an extremely high degree of cephalization, and part of that comes from the fact that when we look at our brain, it will not only be very complex in structure, but also each of the units will have a very specialized function that they in essence have to correspond and collaborate on together. The other thing that we see happening is that we have our spinal cord, which radiates from the brain down the lower back, and when it radiates, what we see happening is from an anatomy standpoint, we are looking at a hollow nerve cord. This is interesting to point out because a lot of times we see that, especially in like invertebrates, the nerve cord tends to be solid. Now, as I mentioned to you before, your nervous system will be divided based on central nervous system and peripheral nervous system. The central nervous system will be our brain and our spinal cord region, whereas the peripheral nervous system will be all the nerves that radiate out of the central nervous system. And the peripheral nervous system is then obviously where you will have all your afferent and efferent neurons, which will be providing your sensory input and your motor output. The other thing that I definitely want to mention, especially since we're looking at this picture right here, is that your spinal cord does not run the entire length of your back. It usually stops around the L1 vertebra, so that's what the little red arrow is indicating. And I do have a little picture that's going to pop up. Let's see if we can get it. Here you go. On this illustration, it's basically also showing you that when the when the spinal cord ends around the L1 nerve, it ends by this area called the conus medullaris, or medullary cone. Now, don't worry that it ends by L1. What we're going to see happening is that we're going to have a radiation of different nerves that will come out of it, almost forming a little horsetail called the cauda equina, as well as the nerves that radiate out from the sides of the vertebra, allowing for your body to basically utilize the nervous system all over, regardless of the fact that the spinal cord ends around the L1 region. Now, if you're interested in learning more about the different nerves that radiate outward, I would definitely recommend that you take a look at table 7.1. So please utilize the PDF of the chapter. And over there in that table, you're going to see a breakdown of the cranial and the spinal nerves. The cranial nerves will be the nerves that will specifically come directly from the skull. There are 13 of them paired together. Now, some tables will only show 12, and that's because there's a little bit of a discrepancy about out if we're going to recognize the nerve that's labeled as 0, because that's the one that will allow us to detect pheromones. And as I mentioned to you before, the debate is still out there whether we're going to recognize it or not. But either way, you'll be able to learn a little bit more about the cranial nerves, so that, for instance, cranial nerve number 1 is all about the olfactory, number 2 is about the optic, the ocular motor is for number 3. So you can go ahead and you can go down your list and take a look at how some of them are afferent, some of them are efferent, and some of them are mixed. And then the spinal nerves will be the ones that will branch out from the spinal cord as they kind of eject themselves from in between the vertebra. And they are usually named based on the region that they are exited from. So you'll have things like the cervical, which is around the neck, the thoracic region by the chest, and then as you move down the spine, you're going to have the lumbar, the sacral, and the coccygeal region. Many of these will be mixed nerves. I will not be asking you the specific nerves on your exam and their function, and I'm assuming that many of us have seen these nerves before, and we have other items to discuss. But for those of us who definitely want to pique our curiosity and take a look at it, I would definitely recommend to go ahead and take a look at table 7.1 to get a little bit more information on the cranial and the spinal nerves. The next thing I want to point out is the difference between gray and white matter, and this all has to do with if we have myelination or not. So I had previously mentioned to you before that your axons will be myelinated, meaning that they'll have a lipid layer that will be wrapping around the diameter of the axon, and this will help with speeding up the electrical signal as it's coming down the axon. Not all of them are myelinated, so what we see happening is that the axons that are myelinated will appear as white matter, whereas the gray matter will be simply the neuron cell bodies that do not have myelinated axons involved with it. An interesting fact to point out is if you take a look at your illustrations on the right-hand side, you're going to notice that you have a cross-section of the spinal cord and a section of the cerebral cortex right over here. Notice how the area of the gray matter and the white matter is flipped. What I mean with that is in our spinal cord, the white matter is located on the outside surface, where the gray matter is towards the center, inside. And in the cerebral cortex, it's flipped. Gray matter is towards the outside perimeter, and the white matter is located towards the central inside aspect. Since we're on this slide anyways, I also want to show you the difference between the dorsal section and the ventral section of the spinal cord, because it's going to be very important. So go ahead and take a look at the top illustration, and I've added a few red boxes to kind of have your eye look at. And notice how I went ahead and I highlighted for you the dorsal horn and the ventral horn. Now, the dorsal horn, that's going to be towards the back. The ventral horn means that it's towards the front of the body, so pointing towards the belly. And what we see happening here is that the dorsal side will be the side where the afferent axons are able to communicate their signal with the interneuron and introduce it into the integrating center. So the back part of the spinal cord, the dorsal part of the spinal cord, is all about receiving signals from the afferent axon. The ventral side of the spinal cord, so the side that's pointing towards the front, towards the belly, that will be all about housing the efferent axons, which means this will be the ones that will be communicating with the effector organs or glands. So this will then be the motor units that will be activated once we decide what our course of action will be all about. The central nervous system, because it is so essential to the body, as you can imagine, there's some extra steps that go into isolating it and protecting it. And one of those steps involves the use of meninges. Meninges are going to be layers of connective tissue that will surround and protect both the brain and the spinal cord. Across the different animal models, we can see different numbers of meninges. So for instance, fish will tend to have 1. Mammals, on the other hand, will tend to have 3. So when it comes to us, if we look over to our illustration, on the right-hand side, we're going to notice that for the cranial meninges, there are 3 of them that are labeled. Starting from the outer to the inner, we have our dura mater. Our dura mater tends to be nice and thick of a connective sheet. Then we have the arachnoid mater, which has more of like a web-like appearance, so it's a little bit thinner. And then the one that is going to be the deepest and it's also going to be the thinnest and the most delicate, that is going to be the pia matter. And what we see happening is that the meninges, the dura mater, the arachnoid matter, and the pia matter will be wrapping around both the brain as well as the spinal cord. And as they're wrapping around, it will create some cavities that will be filled with cerebral spinal fluid. Cerebral spinal fluid will rotate through the central nervous system, and as it's rotating, not only is it filling up the different spaces, but it's also an excellent way to provide cushioning and a shock absorber, because we do see that our brain tends to float around in our cranium. The blood that makes its way over to the central nervous system also has to pass through a blood-brain barrier, and that's one of the ways that we can limit solutes from being transported into the central nervous system. We do know that there are certain small lipid-soluble particles that can easily pass through. This is one of the reasons why alcohol has the effect that it has, allowing you to feel tipsy or drunk, because those molecules are easily passed through the blood-brain barrier. Most solutes will require specialized carrier transports. This is one of the reasons why it makes it very difficult to establish medication that has to go ahead and have an effect on the brain or any part of the central nervous system. The blood-brain barrier often has to be taken into effect if it will allow the medication and its components to pass through. We do know that there are areas that have slightly higher permeability. So for instance, our pineal gland, our pituitary gland, our hypothalamus, these are all areas that are able to produce hormones and secrete it. What we see happening is that those areas have a higher sense of permeability, making it easier for solids to go in and out. Here is the development of the vertebrate brain. So let's go ahead and start our story with the top part, when we're going to go ahead and take a look at our neural tube. This is derived from ectoderm cells, so some of your primary germ layers. And what we see happening is, as it develops, the anterior part of the neural tube will be focusing on becoming the brain, whereas the posterior part will be focusing on becoming the spinal cord. Now, as you're looking at the transition on the picture, you're going to notice that the goal is to get three main regions. You want to have a hindbrain, a midbrain, and a forebrain. The hindbrain, the rhombencephalon, that one will focus mostly on things like breathing and posture, a lot of different reflexes and involuntary behaviors. Whereas the midbrain is where we're going to be able to take our information from the sensory, so like our visual, auditory, our sensation for touch, those different mechanoreceptors. This is the part where the sensory system will be able to coordinate that information, and it will serve almost as a relay center in mammals. And then the forebrain, the prosencephalon, that one's going to have a lot of information for our olfactory information. It will also be where we have a lot of different regulations for homeostasis, including our body temperature, our reproduction cycles, part of our emotional turnout, and eating and sleeping. And probably most important to many of us right now as we are going through this class is our learning and memory aspects that need to be processed by the central nervous system. Here is a nice little comparison of brain size and morphology, and we are basically doing a comparison between different animal models. So we are seeing the birds, the reptiles, amphibians, lampreys, shark, bony fish, and mammals. And what you can see from the picture, everything's color-coded. We see that there are a lot of structures that are the same across these different animal models. The size might vary, but they still are there. So you can see you have the forebrain, the midbrain, and the cerebrum, the hindbrain that's being represented. I went ahead and I highlighted the birds and the bony fish, because a lot of these models show that the midbrain tends to be very small and reduced in size. While birds and the bony fish are the exception to the rule. They tend to have a very large midbrain for their size, and part of that seems to correspond to the fact that they will be utilizing it for a lot of sensory signal and coordinated movement. So it believes that it will help them in their migratory patterns, and obviously orient around their 3D environment as they are moving towards through the air as well as through the water. We also see that there's lots of variation with the brain size when you take a look at your overall body size. So the little graph at the bottom just shows you that usually we have a corresponding correlation between body weight and brain weight. As one goes up, the other one goes up as well. However, I do want to point out that birds and mammals, they tend to have slightly larger brains for their body. For instance, when we compare it to reptiles. So I guess that's a cool fact. But remember, at the end of the day, it doesn't necessarily mean that a large brain equals more complexity just because you have more neurons. It's all about making those synaptic connections that will allow us to go ahead and increase the complexity of the nervous system. So let's go ahead and start exploring these three different sections of the brain. Let's see what kind of components they're made out of and what they contribute to our overall nervous system. And I say we start with the hindbrain and then we work our way towards the front. So the hindbrain is going to be extremely vital for a lot of body processes. It will be where we can utilize our ability to breathe. Our circulatory system will come into place. Regular movement will be combined in this area as well. And we see that it's broken down into three regions. You have your pons, your cerebellum, and your medulla oblongata. For those of us who are more visual inclined, please go ahead and take a look at the picture that's on the top right, and you can go ahead and see the three sections that we'll be taking a look at for our hindbrain. So we could see our pons. Here we go. Here's the cerebellum. And then your medulla oblongata is the smaller part right over here. Now, your pons is often described as a bridge, and part of that is because of its location, that it's between the medulla, the cerebellum, and the forebrain. We see that within the functions of the pons, it helps us with our alertness. It will also initiate sleep and dreaming. The cerebellum, on the other hand, is two hemispheres towards the back of the brain. Most of what we see happening over here is that it's able to integrate some of the sensory input from the eyes, the ears, and our muscles, and then thereby allow it to correspond or communicate with our motor commands in the forebrain. So as you can see on your PowerPoint, I wrote down that it's responsible for motor coordination because of this integration of the sensory system. Something that's really unique about the cerebellum, it will help us with our speech, our emotion, our attention span. If there's damage done to the cerebellum, primarily during childbirth, it can cause cerebral palsy. Patients will suffer from severe tremors if this happens, and in addition to that, we also see that their speech pattern, their emotion, and their attention span tends to be affected, as well as all motor coordination due to the importance of this particular section of the brain. It turns out that even though from an anatomy standpoint, the cerebellum only makes up about 10% of the overall brain, it does contain over half of the neurons in the brain. So it is packed full of neurons. And then our last section is our medulla oblongata, that is towards the back right before we enter into the spinal cord. The medulla oblongata has shown to be able to regulate your breathing pattern, your heart rate. It can help with vasoconstrictions of the blood vessels, as well as overall blood pressure. And like I said, because of its location, it is the perfect pathway for connecting the brain with the spinal cord. And also what we see happening is a lot of crossing over will occur here, meaning that the input from the left side will pass over to the right side for coordination, and the input from the right side will pass over to the left side to determine in essence what the interneuron and the afferent neuron would like us to do. Damage to the medulla oblongata, unfortunately, is fatal, and part of that is because of all the prime and very important functions that it contributes to the overall body. Our next location is going to be the midbrain. Now, the midbrain is severely reduced in mammals, and part of that is because most of the functions that we see in other animal models has been absorbed in mammals that will kind of be relayed towards the forebrain. Now, we do see, and this is what I was mentioning to you before, that when we were looking at our comparison, that fish and birds will tend to have an enlarged type of midbrain, and part of that will help them with their auditory and visual stimulus as they go ahead and to coordinate and initiate some of their behavioral responses. The midbrain can be grouped together with the pons and the medulla, and that is then what they term the brainstem. So if you're doing some more reading about the central nervous system and you see them mentioning the brainstem, it simply means that it's a collection of the pons, the medulla, and your midbrain. The forebrain, that one is going to be divided into four main regions: the cerebrum, the thalamus, the epithalamus, and the hypothalamus. And the forebrain, as I wrote for you on your PowerPoint, is going to be all about processing and integrating our sensory information, and obviously coming up with coordinating behavior to match the input that was received. So the first one is the cerebrum. These are going to be the two large lobes that we have towards the front of the brain. As you can see, we have a left side and a right side. Now, both sides are able to function independently from each other. However, what we see happening is that they tend to work together because they are physically connected by the corpus callosum. So here on the picture on top, you see the little corpus callosum bridge that forms, and you can also see it on the side right over here. And the corpus callosum is basically going to act as a relay station, allowing the left side to communicate with the right side. And the reason I want to point this out is because remember that we talk often about how the sensory system will cross over. And also what we see happening is that the left side of the brain does behave differently than the right side of the brain. The left side of the brain is more about analytical analysis. It's usually very heavy on controlling speech pattern. So the left side tries to communicate with speech. The right side is more of what we call the artistic side. It's more about spatial relationship, perceptual problems. The right side likes to communicate more by either drawing or like an artistic interpretation of what we're looking at. Now, there have been cases where we have severed the corpus callosum and we've cut up the communication between the left and the right side. So the question then becomes, how does that affect the quality of life of the patient? And it turns out that the patient can completely continue on living their life as normal. However, they do run into some issues sometimes when it comes to communicating or expressing what they are able to sense or they're able to see. So I want you to take out about 5 minutes of your time, and I want you to watch the embedded video that I have on the PowerPoint. It's going to be covering the split brain syndrome. It is a little bit of an older video, but it's very important because it's going to go ahead and introduce you to Joe. Joe suffered from epileptic seizures, and what they do is if it's very severe, they will actually go in and they'll sever the corpus callosum so that the seizures cannot spread across the entire brain, causing a lot of neuron damage. The seizure will just kind of be limited to whichever side of the brain it was initiated in, the left or the right side. So in this little video, it will talk about the fact how he had the procedure because he had the epileptic seizures, and he can go ahead and continue on having a normal life. And then what they're going to do is they're going to do a little experiment with him where they're either going to show him pictures or they're going to go ahead and write down the word of certain objects, and they're going to cover the left side or the right side of his, I'm sorry, I don't know if I said left or right, either his left eye or his right eyesight. And they're going to see if it matters which side receives either a written or a drawn out stimulus, if the brain is able to communicate that. So it's a very interesting little video. So I highly recommend, please take out 5 minutes of your day and do watch Joe and his little split brain syndrome video. Another part of our forebrain is going to be the hypothalamus, and we've mentioned the hypothalamus many times before. The hypothalamus, remember, was the one that was producing all the different hormones that it could communicate with the pituitary gland. We know that the hypothalamus plays an important role in homeostasis. It does things like controlling our body temperature. It's our internal thermostat. It also plays a role in our reproduction cycle, our sensation for thirst and hunger, blood pressure, let me see what else I wrote, body weight, sex drive, you name it, and it turns that the hypothalamus tends to have a little bit of control into it. And part of that is because of its integral role that it plays within the endocrine system and its full control over the pituitary gland. Now, in addition to all that, the hypothalamus is also part of the limbic system, and the limbic system has often been called the emotional brain. The limbic system slash the emotional brain is basically a collection of 4 items. We have our hypothalamus, we have our amygdala, we have our hippocampus, and our olfactory bulbs. And all 4 of these items are able to work together. And what we see happening is that it allows us to go ahead and influence our emotions, our motivations, our decisions, and even our memories. And as you can see from the little red arrows that I've drawn for you on the side, all of them kind of provide input to each other, so it becomes a real collaborative effort. One thing that I want to point out, for instance, in the hippocampus, that area of the brain is very specific to converting short-term memory to long-term memory. So for instance, oh my God, this is going to age me, but or date me, I should say. Back in the day, you would actually tell someone your phone number. You wouldn't just text it to them. So if you wanted to remember that phone number, part of what you would do is you would just kind of repeat it in your head over and over again. And when you do that simple repetition step, your hippocampus is extremely important in taking that short-term memory and converting it into long-term memory. Now, I know we're not physically verbally telling people what our phone number is anymore, but in case you're studying and you want to remember a certain pathway or a certain definition, when you just repeat it over and over again, the hippocampus is the one that will help you convert it from short-term memory, meaning that you'll forget it within a few minutes, to more of a long-term memory. And that's because it will stimulate more calcium release in the synaptic connections between the neurons. Your olfactory bulbs we came across when we were doing our sensory system, because they're able to interpret our sense of smell. And something that's really cool about this is notice that how you can receive the information from the olfactory bulbs, and you can go ahead and put it into the hippocampus and the amygdala. And because of this, we see that oftentimes your certain smells are able to be connected with certain emotions. So for instance, for me, I grew up in South America where we don't have central air conditioning, and something that I still vividly remember is when we would come to the United States to visit family members, when we would walk into their houses, the air condition to me had a certain smell. And even today, 20 plus years later, when I talk to my brother, I'll tell him, you remember AC smell? And he'll just look at me, he's like, yeah, I remember that, because every time we smell that AC, it just brings back memories of the fact that we were visiting the United States and having access to central air conditioning, something that we were not used to in our native South America. Another thing that I want to bring up to you is the amygdala. The amygdala is your aggression and fear center of the brain. And there have been lots of studies to see what would happen if the amygdala becomes damaged, or if there's like a lesion in it. And usually what we see happening is that people will lose that sensation of aggression and fear, and they become almost more reckless. There was a study that was done where they went in and they went into prison systems for people who were on death row for horrible crimes like murders, and they basically went ahead and they were recruiting to take necropsies once those people died. And in the brain necropsy, what it showed is that a lot of them showed damage to the amygdala, and they were trying to go ahead and have a correlating relationship between a damaged amygdala and sort of like this relentless ability to do such horrible crimes and not have any remorse for it. They've also done some animal models, and I went ahead and I found some of the videos for you to kind of show you how the amygdala will basically allow us to kind of see the pros and the cons of certain things. And if we see that we can be harmed, it's the one that tells us to step away. And if we lose that sensation of the amygdala, we do tend to be a little bit more fearless. And there's a little video of a rat and some food that I want you to take a look at. Let me go ahead and show you where I linked it. So here on the slide, we have our little links for two very short videos where you have a pre-lesion and a post-lesion video, where meaning the amygdala before it's damaged and after it's damaged, and you can see how the animal's behavior will be different. And I'll actually show you a screenshot of it in just a second so you can kind of get excited to see what the video is all about. But before we do that, I want to show you this little picture. This picture right here with all the different facial expressions was part of a physiological experiment where they wanted to see that people who had damage to their amygdala, how that would affect their interpretation of emotions. And what it showed is that oftentimes people that have issues with their amygdala, they have a really hard time linking expressions to their proper emotions. So they're not able to read facial expressions to see if someone is upset with them, if they're happy, or if it's just neutral behavior. And part of that seems to be under control from the amygdala. Here is a little screenshot of our little pre and post lesion video that I posted for you. When you guys watch it, it's super short. What you're going to notice is, as part of the experiment, they set up a little hallway and they have a little robot cat that's going to play the role as the predator. And then there's a little door on the opposite end right here, and we have our little rat coming through. Hey! And what they're going to do in the first video is they're going to have a rat that has a fully functioning amygdala, and they're basically going to put little morsels of food, and they're going to change the location of the food. Sometimes it will be very far away from the cat, sometimes it'll be really close, and they want to see what the rat will go ahead and do. Will it go ahead and tempt the cat and run towards the food and come back, or will it be too scared and kind of just stay in that one spot? So you'll be able to see the first video with the fully functioning amygdala where it's weighing out its option, and it does have that fear center that can kind of control its behavior. And then in the second video, that video is going to be considerably shorter. This is going to be after a lesion has been inserted in the amygdala and that fear center has been lost. And what we see happening is, this is the screenshot from the post lesion. They went ahead and put the cat as far as they could down the hall, and they put the food very close to the cat. And you'll see how the door will open up and the rat will just go ahead and beeline it for the food, not having any issues with the cat being so close or even getting fearful of the predator's behavior. So it's a cute little video. Go ahead and watch it. I do want to warn you that the pre-lesion video has a really annoying sound to it, so I would definitely mute it before I start watching it. And then one last thing for our forebrain is we have to discuss the thalamus and the epithalamus. The thalamus is a relay center. It is part of the reticular formation, and one thing that it does is that it's able to kind of act as a filter by blocking some of the signals that we're always inundated with. A really good example of that would be sort of like white noise. So for instance, if you're in a room, or let's say hopefully pretty soon we'll be having our parties again, and we'll be having our meetings again. When you're sitting, for instance, at a party and you're talking to your friend, you are able to listen to what she's saying or he's saying and go back and forth, and you're able to tune out all the other noises and conversations that are around us. But your brain is still interpreting all the sensory information. How do we know that? Well, have you ever been in the situation where you're having that one-on-one discussion and all of a sudden you hear your name in the background being mentioned and it kind of perks up your attention? Well, that is all due to the thalamus. The thalamus is the one that's always taking in the sensory information and that's figuring out what we should pay attention to and what we can ignore. So we're going to pay attention to our friends because we're having a meaningful conversation, but if we hear our name, there might be a chance that, someone is calling us or someone might be talking about us and we want to know what that's all about. So the thalamus will definitely alert us from that background noise as well. The epithalamus is located slightly above the thalamus. I just realized I didn't put a picture of that. My apologies. And with the epithalamus, probably the most important item there is going to be the pineal gland. Within the pineal complex, we see that we have the secretion of melatonin and the production of serotonin. Those two hormones are very important for establishing our circadian rhythm. Circadian rhythm is basically when your body needs to be awake and alert and when the body needs to rest and sleep. For many of us, that means that during the daytime when we go to school or when we go to work, this is when our body is flooded with serotonin, and when the body is time to relax, that would be usually nighttime, we see that the body will become flooded with melatonin. Now, let's say that you're someone who actually is more active at night and tends to sleep more during the day. Well, then the pineal gland will adjust, and what we see happening is that the melatonin will be high during the day when you're usually relaxed and you're sleeping, and the serotonin will be at an elevated level at nighttime when you're working or doing your activities. It doesn't always correspond to sunrise or sunset, but people do like to call it the circadian rhythm. If you've ever had jet lag and you've taken tablets or you've had some sleepless nights, some of the over-counter medications will be full of melatonin. I will let you know that the studies are relatively inconclusive, whether they help or not. Some people feel like it does, others have absolutely no effect. But either way, I digress. The pineal gland in the epithalamus is all about the circadian rhythm, whether you're going to be active or you're going to be mellow and relaxed. Now, having said that, I think it's perfect time for me to kind of stop talking so you can take a little break from my lisp. And we will continue on at a later time with part 2 of chapter 7.